Hello students, good morning. Welcome back to a new video of Sunanda's tutorial. In this video, we will discuss about Western blotting. So Western blotting, otherwise known as immunoblotting, is a process of first separating proteins, then identifying them in a complex biological sample from a complex biological sample. So here the polyacrylamide gel electrophoresis is used and which is prerequisite for Western blotting because we can't separate proteins using agarose gel electrophoresis. For proteins, polyacrylamide gel electrophoresis is used to separate different proteins. So the process of Western blotting involves the transfer of proteins separated by STS space to an adsorbent membrane. Then proteins can be identified on the membrane by different means. Western blotting has revolutionized the field of immunology with the use of antibody probes against membrane-bound proteins. So this immunodetection of proteins has a wide application in biochemistry and other fields of science as it can detect and characterize a multitude of proteins. So what are the principles of Western blotting technique? It is an interaction between protein and probes used for the detection of proteins. Proteins used for Western blotting are separated by gel electrophoresis. The proteins are then transferred to nitrocellulose or polyvinylidene chloride membrane where they are immobilized. That means they are blocked. The transfer of proteins to a membrane is called as blotting. The proteins on the membrane can either be detected by using a reporter labeled primary antibody directed against the protein or a reporter labeled secondary antibody directed at the primary antibody. Okay. So what are the steps? At first, sample preparation, then protein extraction, then proteins are denatured and separated using SDS page, then blotting, transfer of that proteins from gel to a membrane, then blocking. These proteins are blocked using a blocking solution to prevent antibodies from sticking non-specifically to the membrane. Then antibody incubation, primary antibody, a specific antibody that recognize and bind only to target protein is applied, or secondary antibody is applied, a labeled antibody that bind to primary antibody is added. Then detection, the substrate is added which react with the tag on the secondary antibody to produce a signal or light revealing the location and intensity of target protein band on the membrane. So these are the steps of Western blotting. Next, what are the applications? Western blotting is an excellent method which with high sensitivity in order to detect a particular protein even when protein is present in very low quantity or low concentration. So it has been used in clinical diagnosis for different diseases. It is a confirmatory test for HIV used to quantify protein or other gene products. So it detects protein by their size and ability to bind antibody. So it is appropriate for evaluating protein expression in cells and further analysis of protein fractions during protein purification. It is also used for analysis of different biomarkers like growth factors, cytokines and hormones. So it is all about Western blotting. I have already discussed about Southern, Northern and Western blotting. So if you liked it, please hit the like button, share with your other friends and please subscribe to my channel Sunanda's tutorial. In my next video, I will discuss another important lecture video. So please stay tuned. Thank you.